Welcome to the deep dive. Today we're tackling something pretty foundational in medicine.
Yeah, what's in the agenda?
How the active ingredients in our drugs, the APIs, actually, form into crystals.
Ah, crystallization. Okay.
We're looking at some of the newer, more innovative ways scientists are approaching this. It's a really crucial process.
It absolutely is. We've gathered some insights on the key advancements. The goal really is to see how these innovations are making things more efficient, more controllable.
And scalable, right, for actually producing the medicines people need.
Exactly. Better processes for better medicines, essentially.
Okay, so let's start maybe with the basics, but with that pharmaceutical twist.
Yeah.
It's not just what the API molecule is.
Right. It's how it's put together, the structure.
We've talked about polymorphism before, haven't we?
Yeah.
That idea that the same molecule can pack in different ways.
We have. And it's quite fascinating, isn't it? One single chemical compound
Your API.
can arrange itself into multiple distinct crystal structures.
Yeah.
Like building different things with the same Lego bricks.
And those different structures, the polymorphs, they behave differently in the body.
Oh, drastically. It impacts things like how easily the drug dissolves, how well it gets absorbed.
It's bioavailability.
Exactly. So getting the right structure is critical for effectiveness and for safety, too.
So the whole crystallization process isn't just about making the solid. It's about making the right solid form consistently.
You've nailed it. And that need for tight control is pushing innovation in how we manufacture these things.
Which leads us to continuous crystallization.
Precisely. That's a big shift we're seeing.
Continuous processing. So moving away from doing things in big separate batches.
Yeah, the traditional batch method. Continuous crystallization offers some real advantages.
Like what? What makes it better?
Well, think about batch processing. You start it, you run it, you stop it. Then you start again. There's inherent variability just in those stops and starts.
Yeah.
With continuous, it's an ongoing flow, a steady state.
Ah, so you can keep conditions more constant.
Exactly. You get much finer, more consistent control over critical things like temperature, concentration, mixing, all throughout the process.
Which should lead to a more uniform product, batch after batch.
Yeah.
Or rather, just a continuous stream of uniform product.
That's the idea. Greater consistency. And we know from, developing dosage forms that consistency is paramount.
Yeah, for ensuring a reliable supply to patients. Makes sense.
Absolutely. Consistent manufacturing means consistent therapy. It minimizes that batch-to-batch variability.
Okay, so that's the big picture process shift.
Yeah.
What about drilling down a bit? Particle size keeps coming up.
Oh, it's incredibly important. Seems like a small detail, but
But it has a huge effect.
it really does. Think about how a drug dissolves. The size of the crystal particles directly influences the rate.
Right, like powdered sugar dissolving faster than a sugar cube.
Perfect analogy. Smaller API crystals generally mean more surface area exposed to body fluids.
So they dissolve faster.
And faster dissolution usually means faster absorption, faster onset of action.
And we saw, I think, when we looked at lipid crystallization, that things like cooling rate can really dictate that final size.
Exactly. Cooling rate is a big one, but also mixing speed, the presence of additives. Scientists can manipulate these conditions quite precisely now.
To essentially engineer the crystals to be the size, maybe even the shape they want.
That's the goal. And because, as you know, dissolution rate is a key quality control test.
Yeah, specified in regulation.
By controlling particle size during crystallization, you're directly influencing that performance metric, ensuring a more predictable therapeutic effect.
It's really about control at multiple levels then.
Fascinating. Now, another area you mentioned was salt formation.
Yes, particularly focusing on things like amine hydrochloride salts, which are very common.
Why turn an API into a salt in the first place? What's the benefit?
Well, many APIs in their, let's call it, native form might not have the best properties for a medicine.
Like poor solubility.
Yeah.
Or maybe they break down easily. Exactly. Poor water solubility is a big one, makes it hard for the body to absorb. Or instability, which affects shelf life.
No.
Forming a salt, like an amine hydrochloride, is often a strategy to significantly improve those properties. Salts frequently dissolve much better in water.
Which helps with absorption.
Right. And they can also be more chemically stable. Makes for a more robust drug product.
So optimize the API. But the outline stressed developing robust and scalable salt formation processes. What's the challenge there? Why is that hard?
Well, scaling any chemical process from a small lab flask to huge industrial reactors is non-trivial. There are always
And
challenges.
engineering hurdles.
Engineering, chemistry. With salt formation, the critical thing is ensuring you consistently get the intended salt form with high purity every single time, no matter if you're making a gram or a ton.
So avoiding other salt forms or impurities creeping in.
Precisely. And that means you need a really deep understanding of the underlying science.
Yeah.
The thermodynamics, like which form is most stable under what conditions.
And the kinetics, how fast it happens.
Yes. How fast the salt forms versus potential side reactions. It requires very tight control over in conditions temperature, again, how you mix things, how fast you add reagents.
It sounds quite delicate, actually.
It can be. Get it wrong at scale, and you could end up with the wrong product or a product that doesn't perform as expected.
And again, connecting back, that ability to make it consistently and reliably at scale is vital for meeting regulatory standards.
Absolutely.
And just ensuring there's enough drug available for patients.
Couldn't be more critical. Regulators need proof of consistent, high-quality production. So developing these robust, scalable processes isn't just good science, it's a requirement for getting medicines approved and keeping them on the market.
It really seems like none of this, the continuous processing, the particle size control, the salt formation, would be possible without some pretty advanced ways to, well, see what's going on. Analytical techniques.
Oh, absolutely essential. Analytical science underpins all of this. You need the tools to monitor the process and, crucially, to characterize the final API crystals in detail.
Like checking for impurities.
Exactly. Techniques like HPLC, high-performance liquid chromatography, are workhorses for that. As we saw in that handbook on impurities, they can detect and quantify even tiny amounts of unwanted stuff.
Ensuring safety and purity.
Yes. But analytical tools do more than just find impurities. They also tell you about the physical form.
Back to polymorphism. Making sure you have the right crystal structure.
You got it. Techniques like X-ray diffraction, various types of spectroscopy, these let researchers peer into the crystal lattice, confirm the exact arrangement of molecules.
So they can verify they've made the correct polymorph, the one with the desired properties.
Precisely. It closes the loop, connecting the process back to the fundamental solid-state properties of the drug.
Okay, so let's recap a bit. We've covered some really key innovative areas in API crystallization.
We have. It's been quite a journey from basic principles to advanced manufacturing.
We talked about continuous crystallization, moving towards more efficient, more controlled production.
Right, aiming for that steady state and consistency.
Then the importance of dialing in particle size, really engineering those tiny crystals for optimal drug performance, like dissolution.
Affecting bioavailability directly.
And finally, the challenges and importance of robust, scalable salt formation, especially for improving API properties like solubility and stability.
Using common examples like epentramine hydrochlorides, but the principles apply broadly.
And underpinning it all, the sophisticated analytical methods needed to monitor, control, and characterize everything.
Absolutely. Can't do modern pharma development without them.
What really stands out, I think, is that these aren't just small tweaks. They feel like significant steps forward.
They really are. These advancements mean we can design and manufacture medicines that are not only effective, but also consistently high quality, which is huge for patients globally.
This deep dive, it really brings home how details that seem microscopic, how molecules packed together, the exact size of a crystal particle, have such a profound real-world impact on the medicines we use every day.
It's quite humbling, isn't it? The level of detail involved.
It makes you appreciate that constant innovation in these fundamental areas of pharmaceutical science is just essential. We need it for progress.
We absolutely do. And perhaps a final thought for our listeners. Next time you take a medication, maybe consider the incredible journey that API took, the intricate science like crystallization that ensures it works safely and effectively. It's a testament to that relentless drive for better health through science.